So my name is Kristen. I'm a professor of silviculture at Northern Arizona University. I first started out in forestry as an undergraduate. I grew up in Montana, and I was exposed to a lot of outdoor activities and could never picture myself at a desk. So I started in forestry and fell in love with it, and then knew I wanted to go to graduate school and chose silviculture and forest health, and I've been intermingling the two of them for the last 20 years or so. The biggest project and what I'll be talking about in the seminar is related to a model species in the southwestern white pine, and we think what we're doing in southwestern white pine can be used for other species, and we're trying to link genomes and phenotypes across the landscape so that we can isolate the genes and the genotypes that might be best adapted for future climate and for non-native diseases so that we can manage the species for the future. We spent years building up with small pots of money from USDA, less than $20,000 at a time typically, to collect seed from these trees, and then we wrote this big NSF grant and we got $4 million. It was a career moment.
Good afternoon, guys. My name is Aaron Whiteheis. I am a master's student with Paula Thornwald at the Rocky Mountain Research Station and Dr. Seth X with the Silviculture Lab in the Forest and Rangeland Stewardship Department here at CSU. In addition, I am an employee of the Colorado State Forest Service and the Forest Inventory and Analysis Program. And I am studying regenerational trajectories within mountain pine beetle disturbed stands here in Colorado. I'm excited to introduce our speaker here this afternoon, Dr. Kristen Waring. Dr. Waring is the professor of silviculture and applied forest health in the School of Forestry at Northern Arizona University since 2006. She received her PhD from the University of California, Berkeley in silviculture and was a postdoctoral scholar in forest ecology at the same institution following graduation. She specializes in finding solutions to a wide assortment of challenging forest health problems using silviculture and other forest management strategies. Her research and teaching have taken her across the western U.S. into Mexico, Costa Rica, Europe, and Indonesia. Invasive species management and climate change adaptation are particular passions, and in some circles she is known as the White Pine Lady for her research on mitigating white pine blister rust and white pine species. With that, join me in welcoming Dr. Kristen Waring.
Thank you. I'm really honored to be here today, and I'm excited to talk to you about my work. I've been a little bit obsessed with white pine since I was a graduate student in Montana. I worked on white bark pine, and then in California I worked on sugar pine, and for the last decade I've been working on the species I'm going to talk to you about today. Part of the reason I'm known as the White Pine Lady at NAU is because of the project I'm going to talk to you about today. So when people mention my name, they talk about this grant and this project. So hopefully you'll find it as fascinating as I do, and hopefully I do the project justice because it's really big and complex. It's a huge collaborative effort. I am the lead PI on our NSF grant, which is where most of our funding has come from. This was a career moment for me several years ago when we were notified from NSF that we were getting $4.1 million to study this species and to do the project, which was really exciting. And so I am in charge of wrangling all of my co-PIs plus a whole set of graduate students and postdocs and staff. And then a lot of collaborators who also help us complete this work. And then we also have a whole set of citizen science volunteers that have been absolutely necessary to getting our project done. So it's really exciting, but it's also been fairly stressful, and the administrative tasks associated with this big grant were larger than I anticipated. But it's very exciting, and so I'm really happy to be here talking about it today. So what we proposed to NSF was that we use southwestern white pine as a model species and look at how southwestern white pine might be, we might do some research to help manage for the species in the future. So I'm going to tell you a little bit about the background of the species to give you some context, and then I'll show you some kind of like hot off the presses results because we're finally in that stage of the project. So southwestern white pine is one of our native 5-needle pines. Most people, even among the forestry crowd, have never even really heard of it or thought about it. Flagstaff, ooh. Does it show up on there? Okay, so I'll just reach. So Flagstaff is in the middle of Arizona, and that's New Mexico next to it. So the black line is the little range map from 1971. So that's the former range map we have. And we refined it about a year ago using our presence data. And so what we know is that we have scattered populations that are on these isolated mountaintops in Arizona and New Mexico. They're separated often by deserts, so this is actually really important to the ecology of the species. And then when you move south of the border into Mexico, it's more contiguous, but it's still a little bit broken up south of the border. So that's where the species grows. It's part of the mixed conifer ecosystem. So if you move into low elevations, it's a little too dry, and it's not as fire tolerant as ponderosa pine, which is the next lower species. And so if you run fire through it, you lose the white pine component at lower elevations. And at higher elevations, it gets outcompeted by the colder tolerant species like spruce fir. So it's a solid component of our mixed conifer species. It's not a foundational species like white bark pine. So if you lose southwestern white pine, you're still going to have a mixed conifer forest. However, being passionate about white pines, I think it's an integral component of our biodiversity. It's also got these really large seeds, so it's really important for wildlife. And it's just a special component of these forests. And I think we're losing something if we lose any species out of our forests. So I think it's important to study even these sort of more minor components. So I'm going to show this little video. Hopefully it'll work. Oh, back up. I might need help to run the video. It worked fine. And maybe I have to get out of this mode. Maybe I can just play it by doing that. Okay. So to give you some context, NAU was really excited about this grant, and so they put together this promotional video, and I think it's just a nice overview of the project to kind of set the stage. It's only about 2 minutes.
Tree climbing women scientists are going to great heights to find genes with the perfect fit for the future.
If I had a phone book, I'd probably just grab them, but I don't, so I'm just going to go up and get them by hand.
Hannah Stapleton is reaching for pine cones at the tops of tall southwestern white pines.
These cones are mostly open already.
Northern Arizona University scientists believe seeds in cones as high as 80 feet above the ground may hold genes tough enough to withstand disease and climate change.
This one's nice and full, so we know that the seeds have completed whatever development they're going to have.
Forester Kristen Waring says a deadly fungus called blister rust is killing almost all of the southwestern white pines it infects.
And we don't have it here in northern Arizona yet, but it is over in New Mexico and it's moving this way. Between the climate change issues and the pathogen issues, these are trees that we're really concerned about.
Using bubble wrap, they've created a mini greenhouse around the pine cones, raising the heat in the seeds while they're still growing.
Out here on a day when it was maybe going to be 75 degrees, then it might be 82 instead in that bag.
Biologist Amy Whipple will be planting the seeds to find the most resilient ones.
But then it gets even more complicated than that, because even if they have certain copies of certain genes that might help them figure out, it matters whether those genes are turned on or off.
Climbers only have a few weeks to gather the cones and perhaps the right genes for the tree's survival before the seeds fall to the ground.
It's beautiful and it's a different perspective and it's playful, but also it's really cool to know that you're collecting data for a really good cause.
Near Flagstaff or inside NAU, I'm Bonnie Stevens.
Oh, sorry. Okay, I'll go back. Okay, so that kind of sets the stage for some of the complexity on our piece of the project. So I'm going to talk about a lot of other components of the project as well. But you heard two different threats mentioned, and those are the ones we've really been focused on: white pine blister rust and climate change. So white pine blister rust infects all of our native five-needle pines, and it's been spreading. And so in the video I said it wasn't in Arizona yet, but it is. It's out in eastern Arizona. It's just not in northern Arizona yet. And we've got monitoring plots out, and we are tracking the spread. In fact, Chris Looney, who's in the audience today, did his master's work installing those monitoring plots across the Southwest. So we're pretty concerned about this because we don't expect to find a lot of trees that are going to be able to withstand the fungus. And if you haven't seen white pine blister rust symptoms in the field, it starts out with this branch dieback. You also see these blisters, which is the characteristic, why we call it white pine blister rust, the spores, and then it creates these cankers and kills anything above the tree, above the canker on the tree. So it is lethal. It does take a while to kill the tree, so it's kind of a slow death, slow and painful. So our question is, where and how much resistance can we find genetically across the landscape, and how do we test for that? And it takes a lot of money to do that. And so that's part of what we were looking for with this grant and the project. And then we're also really concerned about climate change in the Southwest. We're getting much warmer and drier. Even though the precipitation models are variable, with the increased temperature, our soils are going to dry out. Our trees are going to be more stressed. We're already seeing that. Managers are experiencing that. They're reporting shifts in elevation. We're seeing a lot of tree mortality. So it's a really kind of in-our-face threat. And this shows the current distribution of white pine based on climate models and climate species distribution modeling. And then depending on which climate scenario you run, in 2060 we see some range contractions regardless of how dire we think the climate might be. So we're anticipating that the climate niche is going to get smaller for this species. So related to that, we wanted to ask the question of whether there were trees out there in the landscape that were more able to withstand these changes than other trees in the landscape. So our first question was related to the first threat. Our second question is indirectly related to the second threat, find adaptive traits using common gardens. And I'm going to walk you through all these 5 different things. Our third question was really related to looking at the genetic sequencing of the different populations that we're sampling and trying to link our genotypes back to the phenotypes. We're also using remote sensing technology to link genotypes to phenotypes. And then finally, we want to link it all together by modeling gene flow and these adaptive traits across the landscape and linking that to climate change. So ultimately, what we hope to find are some trees out in the landscape that can withstand both the rust and the climate change, and we can manage the species for the future and use that as a model for looking at other species in other forests. So we've been collecting seed, as you saw, mostly with climbers, for the last several years, and we have seed sources from across the range in Arizona, New Mexico, and Mexico. And we sow them in different years because we don't have the capacity, personnel-wise, to sow them all in one year. So we started in 2014 and went through 2017, and we sowed seeds in each of those years and then used those seedlings in a bunch of different common gardens. So I'm going to talk about the different common gardens, but what's important to understand is that we do have seed sources from across the entire range, and coupled with that, we have over 5,000 foliage samples that we're in the process of analyzing for the DNA component of this and the SNP sequencing. So the first question then to look at the genetic resistance testing, this is all being done in Oregon at the Dorena Genetic Resource Center. And the life cycle of the rust, the pathogen, is really complex, and there's a bunch of different spore stages. So what you need to understand is that the ribes, the understory shrub, is the alternate host. And in the fall, the ribes produces spores that infect white pines. And so the folks at Dorena go out in the fall into the mountains of Oregon, and they collect infected ribes leaves. And they bring them back to this garage that they've modified. They have these raised platforms where they lay out all of the ribes leaves with the spores on them. They lay out their seedlings underneath that they've grown for about 2 years. So this is a long process. And then they just make it 100% humidity, and they mist it, and the spores just rain down on the seedlings. So we call this baby tree torture. And they have a lot of mortality that happens up there. We do baby tree torture too in a different way in Arizona. So this is a little faster torture and mortality because we're specifically trying to kill off the ones that are susceptible so that we can find the ones that have some genetic resistance. So after they inoculate the seedlings, they put them outside, and they track them. And in the background here, you can see a lot of mortality that's going to be directly from the rust because those seedlings have been inoculated. So those are all susceptible. But if you were to find one row of green trees in there, that's probably a family that has some resistance. So that's what we're looking for. So these are our southwestern white pine seedlings put out while they're tracking them, and they have very detailed measurements. They go out and they look at cankers, and they count the number of stem cankers. They also look at needle spots. So the first thing that's infected are the needles through the stomates. And you can see that with spots really early on in infection. So multiple times a year we have people out in these gardens counting needle spots, counting cankers. I've been out there, I've helped them. So have other people in this room. It's not easy, it's somewhat tedious. And so one of the pieces I'm going to show you today is a way that we can get around some of this heavy manual labor and make things a little bit more efficient, potentially. So if you're not familiar with the resistance, there's two different types. The major gene resistance confers immunity to the tree, so you're not going to get any infection. So of all those seedlings that we rain the spores down on, some set of them, typically less than 10%, are going to be fully immune, and they're not going to get the disease. The weakness is that this can be overcome by pathogen mutations, so it's not what we want for the long-term resistance and sustainability of the species. What we really want is the multigenic or partial resistance. So this is resistance that the tree is going to actually get the infection. They're not going to die from it. And this is going to be a longer term, more durable type of resistance. So this is what we're actually looking for in the long run. It's nice to have some major gene in the landscape because you know then that the species isn't going to completely die out. And so we're also doing some side projects looking for major gene. And so today, all I have are results from the major gene tests that we've been doing because it takes longer to look for the partial resistance because those are seedlings we have to track for a longer period of time to find the partial resistance because, again, they're going to get infected. There's generally a low frequency across the landscape, although it looks like some of the trees that we're testing have a higher frequency of resistance than some of the other related species like limber pine. Okay, so preliminary results for the major gene. To explain this, what looks like a complicated set of pie charts, if it's red or orange, it doesn't have major gene resistance. And if it's blue or green, there's a greater likelihood. We know that we have this individual over in Bradford Canyon, New Mexico, that has a major gene for resistance. And we use that as a control in these trials, so we know we're getting good inoculation. So we always include a control in these trials. You can see that there's more clustering around here. This is where the rust has been present the longest in New Mexico and Arizona. And so there's a greater chance that we're going to find the resistance there because there's been selection pressure already on these trees. The rest of this is sort of needle in a haystack because most of it hasn't been hit with the disease. So there's been no selection pressure, and yet we're finding needle in a haystack, some potential resistance out in the landscape. We also see a general trend where the more northern populations to the west have none, so around Flagstaff, and then we get more resistance as we move to the southeast. But we don't know if that trend will hold up with time. This is just a more up-to-date figure with different families. So each family is a maternal tree, so they're open pollinated. And if you have major gene for resistance, you would expect that 50% of your offspring will be susceptible because you don't know who the pollen donor is. So Web 50 and Web 48 both come from the same mountain in southern Arizona, and they both appear to have some level of resistance. RAN 88 is also from northern Arizona, and this population is actually from Mexico. So these are the ones that have potential for resistance. The ones on the other side of the 1-to-1 line are probably fully susceptible, or maybe have some partial resistance. Something related to management with the Web 50 and the Web 48 is that in summer of 2017, that mountain burned over, and they're both dead. So there's some urgency in the Southwest. Remember how those isolated mountaintops, if you have a fire that gets going on one of those isolated mountaintops, it can destroy an entire population in one fell swoop. And that's what's happening with some of these populations. So since I started collecting cones from these populations, we've lost maybe 20 of these seed trees already to fire alone. So there's some urgency on the side of both managers and researchers to figure out where these populations are. And when we went and checked last summer, we were prepared to go back in and collect scion and clone them. Unfortunately, they had already succumbed to the fire. But the Forest Health Protection folks are actively out there collecting scion and creating a common garden of scion out in a grassland away from the forest where we think we can protect them better. So it's not all doom and gloom, I guess. Okay, so moving on to the common gardens that we have in Arizona. We use what's called the Southwest Experimental Garden Array. And this is a set of 10 experimental gardens designed to look at how vegetation responds to climate change. So these 10 different sites are across an elevational gradient and 2 different soil types. So if you wanted to study how anything is responding to warmer temperatures, or do a full reciprocal transplant study where you move things from lower elevation to higher elevation and vice versa, you could do that really nicely under the design that I'm going to show you. The whole SEGA experiment was funded by NSF, and anybody can come and pay to play, essentially. And so if you're interested in climate change research, we can talk later about how you might get involved in the SEGA project. I'm going to talk about just 3 gardens. Our high elevation garden is in this aspen mixed conifer zone. Our middle elevation garden is in the ponderosa pine zone, and our low elevation garden is in the pinyon juniper zone. And we hold watering fairly constant, although I'm going to talk about a drought study as well. But we're really looking to simulate warming across these elevations. So remember, southwestern white pine is part of the mixed conifer zone, and so as soon as we move into those other gardens, we're dropping it in elevation for the southwest. However, we have populations from Mexico, which is a much lower latitude, and the temperatures actually start to approach what we find at our pinyon juniper site. So we actually were hopeful that we could bring seed up from Mexico, and it would just do gangbusters at the pinyon juniper site. So this is what these gardens look like. They're all equipped with weather stations, so we have access to real-time weather data, which is a really nice aspect of the study. Sometimes they go offline because they're really remote, but we can always access it later. So these weather stations are constantly collecting data. The gardens are all fenced because we do have both cattle grazing, and then we have bison nearby one of the sites, and so we need to keep the animals out. And then we have an irrigation system set up at all of our sites, and we have rainout shelters, which I'll talk about a little bit more later, on about half of our boxes. We designed these boxes to be above ground. We didn't plant those trees in our native soil because it would have been really impossible. It was hard enough to get these boxes out there and to get the soil and the microclimate all the same across all of our gardens. And so it would have just been really difficult to keep the environment the same. And I was telling somebody earlier that at this particular site, this is our low elevation site, a grad student was doing a soil mycorrhizae study, and he took a bobcat out there with a soil auger on it and broke it in an hour. So I was at that point really happy that we had made the decision to plant in raised bed boxes. So we have up to 25 replicates per maternal tree. So maternal trees are those trees we collect the cones from. And so we plant them into these boxes. We have 100 seedlings per box. until some of them start to die. We've got about 22,000 seedlings out there over our 3 our 3 planting years. We have 75 boxes per garden right now and a completely randomized design. So we take the seeds that we want to sow and we randomize them into each garden prior to sowing, and then we plant them out in that same randomized design. So it's a nice experimental design from a statistical standpoint, but from a logistical standpoint, it was a lot to wrangle. I mentioned earlier we had citizen science volunteers. These are some retirees who spent some of their time out planting. This is not easy work. We were really impressed. They put in really long days and helped us plant. These are some folks who work for either SEGA or Grand Canyon Trust. They've also put in a lot of time helping us put these seedlings in the ground. Once we take the seedlings out of the greenhouse and move them out to the site, we have a matter of just a few days before they start to desiccate and start to go downhill from a vigor standpoint. So it's important that we get a team of people out there working. We've also had rodent problems, so you can see we've had to put flashing around the boxes to keep the mice out of the boxes. They're a continual problem. Even though we have rodent-proofed the bottom, they find their way in and nibble around our seedlings and cause mortality. So it's a series of challenges, and the grad students one summer got really good at using their knives to stab the mice. There were that many out there like running around on the sites, and they're eating the graduate student projects. So life and death in White Pine Country. We have a couple of other treatments layered in on top of this. So we have an epigenetic study, which was mentioned in the tree climbing video, where we climbed the trees in the spring after the seeds were fertilized, but before they were fully developed, and we put bags on them. So here's a bag here that Hillary is holding onto to try and simulate a warming climate and the effect that might have on the final seedling that's produced from those seeds. And that's called epigenetics. And so we did this along an elevational gradient at some of our sites. And then we went ahead and put those into our randomized design as well. So on top of all these seedlings, we have this extra layer of treatment on some of them. And then we also have the drought experiment. So we have rain out shelters on half of the boxes. And we've reduced that rain that comes in to about half during the growing season. So these are plexiglass tubes essentially that just pull the water and run it out of the box so that the seedlings aren't getting that water. And at the same time, we reduce their irrigation water by 50%. We're still refining this because we found that at our high elevation site, because it's so remote and we weren't getting the real-time weather data that we needed, we didn't quite stress them as much as we needed to last summer. So we'll be probably reducing their irrigation water even more and torturing them a little bit more. Okay, so just a quick view of each of the gardens. So this is the high elevation garden in the mixed conifer zone. This is our mid elevation garden in the ponderosa pine. And this is our low elevation in the pinyon juniper. So the next few slides are the results of one of the master's students who's finished already. And she looked at differences in seedling traits and performances across the different gardens. And the remote nature of the site was actually really challenging. So she was trying to do photosynthesis, which involves a lycor, which will only run for so many hours without a battery attached to it from a truck. And we can't even get the trucks close enough to our boxes. And so she was limited by that. And then the drive time between sites also limited her physiology data. And so we ended up just going with some other data because her physiology data was limited. So she collected needles and did heights and diameters. And what she found was that there is this latitudinal gradient. So this is population latitude. So as you move to the north, so low latitude, high latitude, and so the seedlings from low latitudes did indeed grow taller, which is what we thought might happen because they're coming from Mexico. There really wasn't a garden effect, so we found the same relationship across gardens with latitude. Stomatal density varied as well. So the stomates are really important in regulating water use. And so the mid and the high elevation garden were similar, but the high elevation and low elevation garden were different. So the low elevation garden, there were lower stomate densities. So they have fewer stomates in the same families at the low elevation garden, which is pretty interesting. There is a north to south latitudinal gradient as well. It's not as distinct as what we found with height, and there is an interaction between the garden and the population. So it's a little bit messy, but it's interesting that the same seedlings from the same families have lower stomates at the low elevation garden. And when we coupled this with carbon isotope ratios and percent nitrogen, so the low elevation garden had lower or a higher carbon isotope ratio and different percent nitrogen, which was also intermingled with the population effect. But the overall bottom line of this is that they had greater water use efficiency because of the stomate density. The carbon isotopes showed that. So they were able to close their isotopes essentially and use water more efficiently. So they're at the highest temperature site, right? They're at the low elevation garden. And so this is actually a really interesting finding from a climate change perspective. The seedlings are actually able to adapt to that lower elevation. Okay, so from this study, the height was greatest in the southernmost populations from those lowest latitudes. The lowest amount of height growth at the warmest garden site, even though we had extra water. So the growth rate, even though the height was greater, the growth rate in a season was lower. And the low elevation garden seedlings had the lower stomatal density and higher water use efficiency. And so from a climate change perspective, this could allow for greater growth in these low moisture environments. She was looking at the 2015 sow year, which was all US populations. We now have Mexico populations out there, and they're not doing well in any of the gardens. So I'm not sure that this conclusion will stand once we start to analyze data from across the entire range. So it's a mixed result. At the 2015 mortality was really low. 2016, we have a year effect on the planting. So 2015, we did a good job planting. 2016, we had a lot of mortality. We think it was a planting effect. And 2017, so far, it looks like the Mexico populations have really high mortality rates. So they don't like being cold, is the bottom line. Which is unfortunate because we had all our eggs in that basket. We were really hopeful. So an ongoing study with one of my PhD students is looking at those drought treatments, and so he wants to see whether the water reduction will result in differences between the populations. We found this in a greenhouse experiment that Betsy Goodrich did for her PhD. So the seedlings, once they were drought stressed, their physiology changed and their isotope discrimination changed, and so their water use efficiency changed. And so we're anticipating that once we start to drought stress these and see a true effect of the drought, we'll actually see a shift in that corresponds to what we found in the greenhouse. Maybe we won't, but we'll see. And then this population effect, we're expecting to see a change in drought and cold tolerance. So we haven't started the cold tolerance study yet, but we're hoping to do that this fall. We had an equipment failure last fall, and so we didn't quite get it done. And then we expect to find a heavy interaction between the garden and the abiotic climate, just like we have so far in our preliminary results. Okay. So moving on to the genetics work, I promised Chris the big reveal. So what species is it? And I'll give you some background here. Our options include Pinus strobiformis, Pinus fluxulis. If you're north of Interstate 40, it could be limber pine. And if you're south of Interstate 40, it could be southwestern white pine. And the background on this is that this is Interstate 40. It runs across Arizona and New Mexico in the north part of the states. And when I moved to Flagstaff and I asked what species we had of white pine, this is what I was told. Because, of course, the species divide by the interstate. Or it could be none of the above. Should I poll the audience? Okay, so of course the answer is going to be more complicated, and it's none of the above. We're in a big hybrid zone in the Southwest. So this is a paper that came out last year, And Anna Shuttle's been heavily involved in this work. So the green squares up there that I can't reach, those are limber pine. And these brown squares in southwestern Colorado and Arizona, New Mexico, those are all hybrids. So every one of our populations in Arizona and New Mexico is a hybrid. And then you find the true strobiformis down in Mexico. So it's really complicated, and we're still analyzing this data, but it's pretty exciting because we've known for a long time that these species hybridize. We just didn't realize that we were in this full hybrid zone in Arizona and New Mexico, and that maybe we'll find a few individuals of one or the other, but the vast majority of them appear to be hybrids. So linking that with some of the other work that we've been doing, so linking the genotypes with the phenotypes, this is some work that Ethan's been working on, my PhD student, where he looked at early growth traits in the greenhouse, so diameter and height, cotyledon number, cotyledon length, and lo and behold, they cluster just like our hybrid graph. So the hybrids cluster together, and the strobiformis clusters together. And when we started this study, we didn't include the limber pine. So I would assume that the limber pine would cluster separately. And I think that Dr. Shuttle would agree with us, or with that assessment. We've also just recently linked codon and seed morphology across the entire range to the genetics. And so this is kind of a complicated graph, but essentially we looked at cone and seed morphology across the entire range, and then looked at these different factors to see how much variation we could explain across the range. So vegetation variables, climate variables, the genomic underpinnings, and then spatial and topographical variables. And between all those different variables, we were able to explain 73% of the genetic, or of the variation, and a huge portion of that is actually genetic. Interacting with other factors, so these numbers are how much variance is explained in this little box that's also interacting with all these other factors. So it's not a clean, like, it's not just the genetics are explaining this and climate's explaining this. They're all interacting and helping us to explain all this variation. So this is pretty exciting, and I'm excited about it because we're actually now linking the phenotypes and the genotypes, which was one of the points of the study. Okay. So moving on to remote sensing, so piece number 4. We're doing this with both unmanned aerial vehicles and mounted sensors. And this is work that's led by the folks at Oregon State University. And they come out and they fly our gardens about once a year. And then I'm also going to show you what they're doing at Darina with the rest. So the idea here is that they use a visible light sensor, so they're doing a regular picture, and that what we want to do is identify each seedling remotely and link that phenotype to the underlying genotypes. And if we can do that in these garden boxes, maybe we can expand it at some point out to the regular forest. This would save us a lot of time and energy in terms of monitoring some of these species, but it's fairly challenging, as you might expect. And so one of the first things they did was use their thermal infrared camera, and they started at 120 meters above the ground, and then they flew at different heights to see at what resolution we needed to fly, or essentially, or what height we needed to fly to get the resolution that we needed. And the bottom line was that we had to do really low flights to detect these individual seedlings in the boxes, which are pretty small. So 8 meters. And you can kind of see the individual seedlings inside the boxes. And that was enough resolution for them to link it back to our tree tags and our individuals. This is another flight from 2016, and it just shows the thermal imaging. And so one of the things that we took away from this was we have a major thermal gradient inside our boxes. And so now we can account for that. We're already accounting for it in terms of placement of the individual seedlings in the boxes in our statistics. But now we know that it's a definite thermal gradient, and it's actually caused a lot of mortality on the warmest sides of the boxes, which is pretty interesting that they can detect it from the air on the thermal imaging. They told us about that thermal gradient before we saw it on the ground. And then we're also now using the remote sensing at Darina. And so these are those seedlings that have all been inoculated with the rust. And so we have these sensors mounted that are, we can move this rack, and the camera then slides over the image, or over the box, and takes spectral imagery. And this signature of the healthy plant is different from a signature of a diseased plant. So the spectral signal is different. And so you can automate this and detect which of these seedlings are diseased and which are not, which is huge, right? So we've been out there scoring with leaf spots and counting cankers on thousands. That's what all these people in the background are doing. They're scoring these seedlings for rust. So if we can help automate that, it would be actually really huge, and we'd be able to get a lot more testing done. And so here's one of their automated images. So the blue are healthy seedlings, and the red are diseased seedlings. And they found a very high correlation between this photochemical reflectance index that they derive from this image and a healthy seedling all the way up through a dead seedling. So essentially they can use this image to just say which trees are diseased, which ones are already dead, and which ones are healthy. So this could really help us. And we just got a grant from the Forest Service, the STDP program, to continue this work and build this piece of the project. So that's really exciting. With climate change, we're slowly building that piece. So this is towards the end of the project. This is a paper we published last year looking at the impacts of climate change on that new range distribution map that we developed. And I know it's kind of hard to see, but we ran 2 different climate change scenarios. And what we found was that by 2080, with just the most extreme model, there was range contraction in the orange areas. With both the less extreme and the extreme, we found contraction in the red areas. So more range contraction expected in the southern parts of the range with the more severe climate change model, as we would anticipate. And a northern range expansion, so all the blue areas are where we would expect the range might expand given climate suitability for the species. And a lot of activity in the hybrid zone, which is really exciting because now we know that's truly a hybrid zone. And some of the most current work coming out of Virginia Tech that we're about to publish indicates that that hybrid zone is actually moving north. So as the climate shifts, we're starting to already see that the genetics are disjunct from the morphology of the cones and seeds, which is pretty exciting from a science perspective. Okay, so to come full circle back to just the last few slides, I talked about finding genetic resistance to the rust. I talked about the common gardens that we're using in Arizona. I talked about the genetic testing for the sequencing and the hybridization that we found. We have a lot more sequencing to do. And I talked about the remote sensing technology. And then I talked about some linkages between these areas. And so the final piece that we'll really be focusing on the next couple of years, in addition to continuing work on these other components, is really doing this landscape modeling. And Sam Cushman is a scientist with Rocky Mountain Research Station. He does a lot of landscape genomics modeling. And so he'll be leading up that effort. And hopefully at the end then we'll find some of those sweet spot trees where we can really focus in on management. So ultimately our idea is that if we have healthy trees out there, we can have healthy landscapes. And I like this picture because it's from the Guadalupe Mountains in Texas, which is our probably driest site in the US. I've seen sites in Mexico that are equally as dry. But you essentially come out of the desert and you come up and you hit this plateau and you're in pinyon, and then 10 feet later you're in mixed conifer. And it's just such a distinct line. These trees are really on the edge, and we really need to find some of those adaptations so that we can actually sustain these species. And with Guadalupe, a lot of it burned a few years ago. So we have this major fire issue that complicates things in the Southwest. So in terms of management implications, we don't have anything really concrete yet because we're still midstream, but I think this will help us prioritize silvicultural treatments, so things like fire hazard reduction treatments, fuel treatments. We have to be able to get in there and reduce the density so that surface fire can happen instead of crown fire. We also need to be able to do some what I call crop tree release, which isn't the traditional use of the term, but we need to be able to maintain the vigor of those desirable individuals that have the good genetics that we want, the desirable package. And then from a genetic conservation standpoint, we really need to be able to focus on those areas that have what we consider the best genetics, but we want to maintain genetic diversity across the species. And then reforestation decisions are actually huge. So we have seed zone considerations. Right now, if you just go by strict seed zone boundaries, I mean, things are changing in that realm, but seed zone considerations do not include blister rust resistance. They don't include adaptations to drought, except for in a really broad sense, and only when you talk about changing up how you think about seed zones. So this is going to dictate what to plant and where we plant it, and we can no longer afford to just be planting by seed zones. We have to consider all of these underpinning genetics of where we're going to plant things and what's appropriate to plant in different locations. So I would argue that if you just went out and collected from this tree and you don't know whether it's resistant to rust and you don't know whether it's adapted to the future climate, maybe you shouldn't be planting it. That's a lot of time and energy into reforestation that is probably not going to go anywhere. And so in a lot of these areas, we probably should be focusing on natural regeneration, which is something that a lot of people have been advocating for a long time with the white pines. Okay, that's it. Thank you.
Can you talk a little bit about this species, the populations in Mexico? Like, is it a dominant component of those ecosystems, and is it not?
No, it's really similar from what I've observed out there. I haven't been to all of the populations, but my observations is that it's a component of mixed stands, just like in the U.S.
Is it important for specific wildlife in Mexico, or is it used for timber?
In general, it's a wildlife because the seeds get even bigger. I have some seeds with me. Oh, I knew that was going to happen. Okay. So I'll just pass these around. Please don't take them because they're from Mexico. And I'm responsible. But you can compare them. This one that says KMD is from the San Francisco Peaks right outside of Flagstaff. And these are from Mexico. The seed difference, they're all huge. We can tell, even though we randomize our trials in the sowing, we could tell when we were planting Mexico seeds because they're so much bigger.
Sorry, but these ones are these are the hybrids?
These are hybrids, okay. Yeah. And those are the regular southwestern white. And so from a wildlife perspective, these are really big, nutritious seeds. And so unfortunately, they haven't done a lot of work on the wildlife value, but there's also a lot of debate on hybridization of white pines in Mexico. And so we actually think we have some hybrids in the Mexico lots. Hybrids between strobiformis and something else, or something else entirely. So it's a whole species complex. Yeah, exactly.
Thank you.
Thank you for that talk. It's a great talk. So it's really interesting how that we're kind of in that sweet hybrid zone right now. And I'm not a geneticist, so I'm not really privy to all the ins and outs. But is there any way to take any of the work that's being done on limber pine and perhaps in terms of like white pine blister rust resistance and apply, like, I don't know, like gene splicing or hybridization, selective breeding with resistant limbers? Is that something on your horizon at all? Is that even possible?
Well, they certainly hybridize. I think we haven't gone there, and I welcome Anna to say anything about this, but they have two different major genes, CR3 and CR4. And so since they both have a major gene for resistance, we have something to build on. And we do have the CR3 gene in the hybrid zone. So I don't know. What are your thoughts on that, Anna?
I think it's definitely a possibility in the future. Certainly we could try to, double load, some individuals with both major genes, assuming they are different. We actually aren't positive. But we think they are. So that would be a great thing, because if they were actually different, then a different mutation in the rust would have to overcome them. So you'd have kind of a double protection there. As we get more, a better understanding of the partial resistance, this, the polygenic resistance, that might be harder to transfer with a cross between the species, but it certainly might be in the cards, because limber pine seems to not have a lot of partial resistance. So we might be able to snag some from southwestern white. So I think it's definitely, it's just a little early in our understanding of how everything works, but there's certainly doing the crosses, having either species be the male or the female, it doesn't seem to matter, they cross. So it certainly would be a potential solution in the future if we were all willing to to mush the species that much and be that intensive for these, that this intensive in terms of management. So.
Did that?
Yeah, absolutely. Thanks.
Seth?
So, what's the function of the major genes?
Oh, that is the question of the last 2 decades, probably. So we think there's probably a trade-off to having that gene, but we haven't been able to. I mean, there's so few trees out there with it that we've identified that we've had problems getting a big enough sample size to really look at what trade-offs might be there. Some of Anna's work and maybe some of the white bark pine work has indicated that there's a relationship with cold tolerance. So when we do that this fall, maybe we'll see a relationship with what they find in Oregon with the resistance, either partial or full. But yeah, we don't know the answer to that yet.
It may be early to ask the question, but you mentioned that this could be a model system, that once we understand a lot about southwestern white pine, maybe that'll help with other things. Any thoughts yet on how that might extrapolate to other species or other cases?
We have a growing number of invasive species problems, and so I think the way that we've coupled everything together all at once, I mean, it requires a huge research investment, but I feel like we've made a lot of progress in just a few years because we were able to get that investment. And so my hope is that if we can show we can be successful, then maybe somebody else can make the same case for their system.
So the model of value would be in the way you attack the problem, not so much in the insights you get from the work.
Well, potentially from the insights as well, but with the invasive species, a lot of them don't have any resistance that we know of, or I mean, some of them, they're ahead of where we are with resistance, like chestnut, they're out planting things, and we lost that species before we were on top of it. So it's really hard, because when you talk to managers, their biggest complaint about things like seed zones is they want more information and more research, and they're never going to have a full set of information. And so I hope that we can convince them from some of these studies that it's not just southwestern white pine. It's all these other species are also in trouble. I don't know. That's probably not a satisfying answer.
I'm not judging.
Yeah, so I'm really curious about your epigenetic study, and specifically if you have any preliminary results about the relative role of genetic versus environmental conditions that the seed was developed in conferring drought tolerance.
So we don't have any results from the drought part of the study yet, because we just started that 2 seasons ago, and the data hasn't been analyzed yet. But we did a pilot study with just 200 seedlings in 2014, and those seedlings were visibly smaller, and then we were able to quantify that. So they had less leaf area, essentially, and they didn't do as well. The germination rates are terrible. So we're working on a germination paper right now, a seed study, and I think that that's going to come out as one of the bottom lines with the epigenetics, is that we killed the seeds. Or most of them. I mean, some of them germinated, but the seeds were so small. The development was just really poor. And so it might be that we heated them too much, but it was only a few degrees. And so it's within the realm of possibility with climate change. And so it's actually a little bit scary. Although, if you can germinate and survive, having less leaf area might be good. So you might be more resilient once you germinate. Chris?
Yeah, I guess just to change the tack a bit, we're just at this workshop in Minnesota where we're trying to convince, or maybe, I don't know, they're mulling over the issue of reaction to stands, and people are saying it's a doomed species and we should just remove it all. So what have you found is the best approach to maybe convincing people of the value of these apparently doomed species?
Repeated conversations with the same people. I preach every time I see a prescription that has a species removal preference and white pine is near the top. I'm like, why are you doing that? Like, you know, just asking them questions like, why are you trying to remove this? It doesn't have great commercial value. We need it to reseed the site. We need that genetic diversity out on the site. And they come around. So I've seen it, those species preferences have actually, for the most part, gone away. Partly because that's a shift in how they're writing the prescriptions, but partly, I think, because we ask so many questions, not just myself, the pathologists that we go out with, like just asking those questions and them not having a great answer. Why do you remove white pine before Doug fir? There's no solid reason to do that. So, but I have had the same conversation with the same person more than once.
Do you think that your results would change if you had not raised beds, but like in the actual soil?
Potentially. Ultimately, what we want to do is pick, if we get continued funding, we want to take some of these seedlings from both poor genetics, or what we consider poor genetics, so susceptible to rust or not drought tolerant, plus some of what we consider the best, and actually put them in native soil and see what happens in the long run. Because these garden boxes, they're pretty artificial. You know, we did inoculate them with mycorrhizae from native white pine sites, and we created a soil media that we thought would work well for them, and it seems to have worked pretty well, but we recognize that it's a fairly artificial site. One of the most ironic components of this study is this is our mid-elevation site. At our high elevation site in the mixed conifer on the north rim of the Grand Canyon where we are, there are no white pines. And so I actually don't want to leave them out there long term, too long. If they start producing seed, then we're changing the ecosystem. We don't know why there's no white pine out there. But.
Does anybody have any more questions?
What was the multispectral sensor you guys were using on the frames?
I would have to ask about that because they've changed sensors. So they went from the one that's on the drone is a different one from the one that they're using on the hanging platform that they just roll across the garden. So if you want afterwards, I can get your contact information and I can get you that information.
Thank you.
Mm-hmm. Do we have time for more?
Yeah, we can go to 4:30.
Sure. I was actually curious about the drones. What's your experience with them? Do you think there's a lot of utility to be found? Do you think the technology is close but not all the way there yet? Because it seems like a really incredible tool for surveying, you know.
Right. So one of the problems is that we had to get really low to the ground to get the resolution. So I don't know what that translates to out in the natural forest to try and distinguish, because then you've got mixed species as well, because it's not just white pine. So can you tell different white pine phenotypes and different trees species from each other? The other problem we ran into was that a lot of those sky islands in Arizona are actually military. And that was just a flat out, no, you're not flying anywhere near our military zones. We also, this site isn't so close, but the high elevation site is about a half a mile from the Grand Canyon National Park boundary, and they said, no, we don't want you near our, even though it's a fenced site and we're flying at 8 meters above the garden boxes, they didn't really want us flying that close to the park boundary, and we're not even that close to the rim at that point. So there's a lot of rules that they're still working out with the drones. Sorry, unmanned aerial vehicles. I promised them I wouldn't say that. I think that it's promising. The other issue is that with our boxes, they found that because they had to get so close, they were better off to just roll them on the platform instead of flying them. So depending on the situation, I know that they've had a lot of luck in Hawaii looking at rapido high def and flying and seeing stress trees from the air with the drone. So I think the potential's there. We're just, with the thermal imaging, I think we're not there yet. The multispectral, I think we're a little closer.
Okay, that concludes the presentation. If you guys want to stick around, we'll have food and snacks in the back. And Dr. Warren will be here to answer any more questions.
Thank you.